Yes, so my name is Jack Madsen. I'm the CEO of Diagonal Bio, and I'm here to tell you about our exciting journey to develop quick, accurate, and cost-efficient point-of-care diagnostics. But actually, our technology can also be used for identification of genetic markers in the less regulated market segments, and I will get back to that during the presentation. First of all, I would like to start with this quote from the high-impact journal Nature, which states that every single time we have an outbreak, we are always one step behind because we do not have rapid diagnostics to detect a new organism. And I think that was pretty evident during the COVID-19 crisis. And there will be other pandemics to come. We, of course, with our technology, have a solution for this problem. But before I go into the details, I want to talk a little bit more about the problems with today's diagnostic testing. First of all, a lot of these diagnostic tests, they are outsourced to external laboratories. My guess would be that everybody in the room here have actually tried this during the COVID-19 crisis. So take the PCR test, you go to a test center, and then it's sent to a centralized laboratory. That means long response times, delayed treatment, and risk of spread of infections, but also a chance for complication for the patients because you do not get treated immediately. There's also a lot of sample handling involved in this, which means there can be a lot of rerun on the samples. So we talked to some of these centralized laboratories, and in some cases they need to rerun 10% of the samples, again delaying further the diagnostics. You also need to have this complicated and costly logistic, shipping samples back and forth between the laboratories. And then you need expensive equipment and complicated equipment. They use robotics to purify the samples before they're loading them onto big PCR machines. Of course, there is alternatives. And again, my guess would be that everybody here knows this. You know that there's the quick test or the lateral flow test, as they're called. They're very easy to use, also at point of care. They're cheap. Unfortunately, they're also highly unreliable. All the studies that I've seen have showed that the accuracy is below 50%. So really, there's not really any alternatives if you want accurate results. You need to use the PCR-based technologies. There's also another problem, and that is that most of these diagnostic tests, they're very diagnose-specific. And again, using COVID as an example, if you had respiratory problems, you go to the test center and you get a very binary answer, yes, no, you have COVID. If you don't hit it right the first time, you will need to go back and get another diagnostic test. This also goes for other types of diseases. And we can actually take this a little bit further, because this is also something we see in the less regulated markets, so outside the in vitro diagnostics. This could be for diagnostics of during animal production or in crops, testing for salmonella in food or feed, and so forth. And therefore we have decided at Diagonal Bio that we want to split our technology into two different products. One that's called Lamplify for the less regulated market segments. And then, of course, we still have Panviral that we want to bring to market for the in vitro diagnostics. So how is it that we are so much better than what's on the market today? First of all, our device is a small, neat little box, basically half the size of a laptop. Of course, it has more depth. Has a very easy-to-use touchscreen. And as you can see here on the cartoon on top, it uses a cartridge which has 8 channels, which means we can scan either 8 samples, 8 patients, 8 animals, or whatever it is that we want to scan. We can do that 8 in one and the same run. Or we could scan 1 patient for 8 different diseases, giving a more holistic view on the disease area that we want to analyze. Again, using COVID, instead of getting these yes/no answers, we would be able to say potentially which type of COVID, whether it's influenza A or B or respiratory bacteria. But we can also use this in other areas. Antimicrobial resistance, for example. Treating patients today at the hospital for sepsis, use broad-spectrum antibiotics to be sure that you actually hit the right one. With our technology, you know exactly from the start which antibiotic to use. Our technology is extremely fast. So we have done several studies where we have shown that we can detect the genetic markers for COVID, for example, below 10 minutes. But we also tested RS virus, influenza A and B, and all of these are around 10 minutes. And that means the whole process for actually taking the sample to get the results will be within 10 to 15 minutes. We still have the same accuracy as we have the PCR-based systems, so around 89 to 100%. We can do parallel testing, as I explained, which means that we have this holistic view, whether we want to test 8 patients at the same time, or test 1 patient for 8 different diseases, or test food, feed, or whatever it is for 8 different genetic markers for toxic bacteria or something like that. We can do that with the system. It's extremely flexible. If you want to test for, let's say, a new genetic marker for a new disease that's coming up, it can be done in something like a week. Then we can scan for it with our device. It's extremely easy to use. So the operator will sit in front of the device. And again, if I use COVID as an example, there's something that's fresh in our minds. You would take the swab, put it into a lysis buffer, load it into the cartridge, put it into the device, press the touch screen 3 times, and within these 10 to 15 minutes for the whole process, you will have your result. And because of the technology, it's also cost efficient. We will be able to compete both in the, you could say, high pricing segments, but also the low cost segments where we will find the quick tests. A little bit too fast. This is an example of the data that we're generating with our technology. Here we have actually identified, we have primed the system to look for a genetic marker for COVID. What you see below the orange threshold, that is negative COVID samples. And as you can see here in the positive COVID samples above the orange threshold, it's a crystal clear, easy to read signal. This is the type of data we're generating with our technology. A little bit about our technology without going into too many details. We are using a combination of 2 different methods. The first one is called loop-mediated Israel amplification. And to make this very basic, what we're looking for is a specific genetic marker in genetic material. This can be DNA or RNA based. The origin from this can be either human, plant, animal, virus, bacteria. The system doesn't care. It looks the same for the system. Then when we have found that genetic marker, we amplify it exponentially. And by coupling it to electrochemical sensors, we can detect that very early on and very precisely. And that is actually what we have protected with our patent. We currently have protection on the patent until 2041, and that is of course the whole foundation for our, you could say, commercialization. And I also want to remind you again, we can use this technology in multiple market segments, not only in the diagnostic segment, but also in the less regulated market segments for identification of genetic markers. Then you could say, why is speed then so important? This is a study that was done between Harvard and Ohio University back in 2021. They used COVID as a model. And here it's simplified a little bit to make it easy to understand. The gray bar represents the control group. So if you do no testing at all, you will have 100% spread of the disease into the group. Then you can say that then the more frequent you test, you can of course reduce infections. That's, you can say, a no-brainer. That's actually what you can see here. So even if you test every 3 days, if it takes more than 2 hours until the patient gets the diagnosis, you still have up to 45% spread of the disease into the group. If you, however, testing every 3 days can get the results in real time, basically when you're sitting at the test facility, then you can limit the spread of infections to almost 0. And that's exactly what we can offer with our technology. We offer something that's both quick and accurate for identification of genetic markers, whether that is for used in diagnostics for detecting infectious disease, or for detecting salmonella in eggs, flu in production animals, or whatever it is. We can do this very quickly and accurately. It's a simple one-step process. The operator will be able to sit in front of the device, don't have to move away. Take a swab, for example, in case with COVID, load the cartridge, press the touchscreen 3 times, and you have the result. And again, we have this opportunity where we can actually scan for 8 different either bacteria, diseases, or whatever it is at the same time, giving a more holistic view on what's actually happening. So essentially what we're doing is that we bring in the gold standard of diagnostic testing or the identification of genetic markers down to something that's called point of care, so near the patients or near where you're actually taking the sample. This is a study we did with one of our university collaboration partners where we took samples from patients that was infected with COVID. They did a nasopharyngeal swab, then the sample was purified, and at the university they actually did RT-PCR, so that's what you can see in the bottom of this graph. On the x-axis, that's cycle number on an RT-PCR machine. We tested the exact same samples with our technology, that's the y-axis, and that's minutes it took our device to detect these as positive. For the RT-PCR, you still have to remember that normally you would need to ship those to a centralized laboratory, then you purify the sample before you run them on the RT-PCR system. But even if you compare to what the time it takes on an RT-PCR system, with an optimized protocol, and again this is taken out of the high impact journal Nature, the average time would be around 105 minutes. The average time it took us with these 30 samples was 8.6 minutes. So already here we are 10 times faster than the PCR technique. But then you have to remember again, we don't need to ship samples anywhere. We can do it straight next to the patient. And we do not need to purify the sample either, which basically means that we will be around 250 to 500 times faster than you can say the gold standard of the way that we are testing today. And that is quite unique. And then you could, of course, challenge me and say, Jack, this is something that you did in your own lab. Can we really trust this? And I would, of course, say that everything we do at Diagnos, we have high integrity, but I would agree with you. Let's get somebody else to test this. So as Peter said, this is the first time we showed this data. We took our device to Center of Diagnostics at the Danish Technical University. They used to test 12,000 COVID samples per day for the Danish government during the COVID pandemic. And then they did a comparison between our system and the RT-PCR-based system. And to not make it too complex here, what you can see here on the figure A is in red dots, that's the time it took our technology to detect different dilution series. On the green dots, we have the RT-PCR technology. And again, the axis here on the left side, that's for the RT-PCR system, and that's cycle time, it's not minutes. Whereas on the right side, it's minutes with our system. And then we do a correlation of the two curves. And as you can see here, it looks pretty identical. It's almost a straight line. So what does that mean? It means that our technology delivers the same quality of data as you would do with the RT-PCR-based system. And not only that, the average or the best time that it took for us to identify a positive sample here was 8.5 minutes. If you compare that to the RT-PCR technology that they're using at the Center of Diagnostics, the method they have accredited, it's 3 hours and 20 minutes to run that on the RT-PCR machine. And again, that does not include taking the sample from the patient and shipping it to the centralized laboratory. So this is a really great study. And what makes me really, really proud is that the head of center actually had a quote. And you can read it here. Diagnos technology is easy to use and deliver results that are comparable to the general RT-PCR technology that's used today. And not only that, Helen Larsen, which is the head of center, said that she sees great potential in different market segments with our technology as soon as we get it to market. So this was really a great study, endorsing everything we have said from the start. A little bit about the market opportunities in the different market segments, and this is something we did together with Dr. Astrid Fribjerg. So what you can see here is that for the less regulated market segments, where we plan to produce Lamplify, this is currently valued at around $37 billion US, whereas the in vitro diagnostic segment is valued at around $126 billion US. So in either case, both of these segments are highly valuable. The way that we plan to go to market is that we want to have the main driver of the revenue from the consumables. That means from the cartridge, which is a single-use item, and also for the reaction mixes, which will be specific for each genetic marker that we want to identify, whether that is for salmonella, infectious disease, or whatever it is. And we plan to start commercialization as early as possible. Now that we have something tangible, we have a physical machine we can send out to different laboratories. We have endorsement from a key opinion leader. Talking about out-licensing deals and partnerships are going to get much easier now. And we plan to commercialize early via Lamplify, a system that is intended for the less regulated market segments. Just before I wrap up here, I just want to emphasize the focus and key milestones moving forward. We are focusing on two systems, one that's called Lamplify for the less regulated market segments that will be easier to bring to market quickly, and then we have PanViral, which is intended for the vitro-diagnostik market segments. That will take a bit longer. The most important milestones for 2023 will be to have external validation of the system. And as you have seen this study from Center of Diagnostics, we are already well on our way here. Second part here is that we want to have CE marking of our system to make sure we have a commercial system we can start selling. And getting the first revenue will of course be a super important milestone to reach for us. All of this is of course only possible because we have a very dedicated small team working very hard every day to actually make sure we get all the way. And of course we also have strong collaborators and partners to help us. And we also have a strong and professional board, being a listed company in Nasdaq First North. So with that, I will have the last statement here, and that is to just say that our vision will still and continue to be to limit the spread of infectious disease worldwide by creating an accurate, cost-efficient, quick and universal diagnostic platform and bring that to market, and that's the system we call PanViral. So with that, I would like to say thank you, and I will be ready to take some questions.